Good morning, I'm Meshack Kodiko, teacher of biology. Welcome to our today's lesson. Today we are going to talk about inheritance of ABO blood groups in human. So in our previous lesson we had talked about complete dominance, we talked about incomplete dominance. So today we are looking at what we call codominance, where the two alleles codominate so that they express themselves in an offspring phenotypically. Blood groups in human beings are determined by three alleles, that is, we have A, B, and O. So an individual can have only two of these genes. Maybe you are wondering why an individual can only have two, yet there are three. Genes A and B are codominant, while gene O is recessive to A and B. So what do I mean by that? That gene A is dominant, B is also dominant. So that if we have AO, for example, that person is going to be said to be blood group A because O is recessive. And we already know that a recessive gene is a gene that is not able to express itself phenotypically. In other words, it is suppressed by the other dominant genes. So when we have AO, A suppresses O, therefore what can be seen is only A. The same to BO, we can only have blood group B because B suppresses O. So, because these genes are determined by more than one allele, so that we refer to them as multiple alleles. So ABO blood group is an example of a characteristic that is determined by multiple alleles. Now, the ABO blood group system, we also have to look at what we are calling the rhesus factor. But before that, let us have a look at this. When we are looking at a blood group, we have a group, we have what we call antigen, and what we are calling antibody. So a blood group is determined by these two. When we have a blood group A, an individual blood group A has got antigen A and antibody B. A person with blood group B has got antigen B, antibody A. Then we have individual with blood group AB has got both A and B antigen but lacks antibodies. A person with blood group O lacks antigens but has got both antibody A and B. So you can now see when we are having these individuals that the antigen and the antibody that determines the characteristic, or rather the blood group of an individual. So that is there. Then we have what we are now calling the rhesus factor. Rhesus factor, in addition to the antigens, we also have what we are calling a rhesus factor, which is responsible for the presence of protein antigen D, so in the red blood cells. If blood from a rhesus positive individual or a person is transferred into a person without the rhesus factor, that means the individual with rhesus negative. So the recipient's body produces antibodies against the rhesus factor. But if we take, for example, if a rhesus positive blood is transfused to a rhesus negative individual, then the offspring will be rhesus positive. Why is this the case? It is because the rhesus position of rhesus factor makes it become dominant. So it dominates or suppresses the rhesus negative. So when that one happens, then the recipient's body produces antibodies against it. This will cause what we are calling agglutination of the red blood cells, which can be fatal if subsequent transfusion with blood group is done. So in that case, that is why we are sometimes finding that people may be having their first children surviving, but the subsequent children may not survive because of this antigen-antibody reaction interfering with the fetal red blood cells. Because it affects the fetus, then we call it erythroblastosis. So the disease is called erythroblastosis fetalis because it is affecting the fetal red blood cells. Let us look at sex determination in human beings. Human beings, we know we have the human males and we have the human females. So when we have XY type, e.g. in human male, in males, two types of sperms are produced. Half of them contain X chromosome and the other half contain Y chromosomes. In other words, they are heterogametic. That means that the X and Y are not identical. So because they are gametes, we call it heterogametic. During fertilization, only one sperm fuses with the egg. So if it is an X chromosome carrying sperm, then a female zygote is going to be formed. But if it is the Y chromosome carrying the sperm, then a male child is going to be formed. It then follows that the chances of getting a boy or a girl is half, or what we are calling 50-50. There is equal chance of getting the two. Let us look at what we are talking about diagrammatically. When we have a male and we have a female here, we have said that males are heterogametic. That means that XY. Then we have the female which is homogametic because the gametes look alike. So during segregation, we find that the X chromosome here will segregate Y. Then we have the X. We have X. So when this X fuses with that, a female child is formed there. When we have that, another female child is formed. When we have this, we have XY. Then we also have another XY or YX, whichever. So you can see here that we have 2X genetic ratio. We have 2XX to 2XY. When you simplify this ratio, we are going to have 1:1. So the probability of having a female child is 1/2. Probability of having a male child is 1/2. So it is a matter of chance that any time maybe the fusion takes place, the probability of having a male or a female child is a matter of chance, and it can be 50-50. That's why we say that it follows then that the chances of getting a boy or a girl are half. So let us look at now what we are calling linkage. So the term linkage describes the situation where genes or certain characters are located on the same chromosome. So by the mere fact that they are located there, like we can have X having, let's say, H for hemophilia, then we have a Y. Y does not carry hemophilia. Then we can have a female character which is X, H, X, that being a carrier. So this linkage here, the mere fact that is located on that chromosome is what we are calling a linkage. Now, offsprings produced by sexual reproduction show only parental characteristics and only sometimes few new recombinants. That is, offsprings with combinations of characteristics not found in either of the parents due to crossing over in the first process of first meiotic cell division. Genes are said to be linked when they are located close together on the same chromosome, what we have seen here. So this is a linked gene, such that they are always inherited together. Let us have a look at what we are calling sex-linked genes now. These are genes which are located on the sex chromosomes, and we only know that there are two sex chromosomes. There is either X chromosome or Y chromosomes. So sex-linked genes are genes located on the sex chromosomes and are always transmitted together with those characteristics. So sex linkage refers to the carrying of the gene on the sex chromosome. Gene for a trait may be present, yet offspring does not show the trait. So that means that this happens in human females, XX, where the gene for the trait is recessive. So when it is a recessive one, it means that it's not expressed. The female acts as a what? As a carrier. So, like when I was talking about in the case of hemophilia, when we have X capital H which is linked on it, then X small h, these are heterozygous, but this female is a carrier. But this male is normal in that case. So, because hemophilia is a trait that is not carried on the Y chromosome, most of the characteristics that are carried on the Y chromosome are characteristically male, but females do not have the Y chromosome, and the hemophilia is not carried on the Y chromosome. So you can see that in humans, for example, the sex-linked characteristics are found on the X chromosome. They include the following: hemophilia. What is this that we are calling hemophilia? Hemophilia is a disease that affects the rate of clotting of blood, leading to excessive bleeding even from a minor cut or very small cut. So there is always excessive bleeding that can lead to a person losing a lot of blood and very vital nutrients from that blood. So hemophilia is more common in females than in males. Maybe you can ask a question why hemophilia is more common in males than females. This is because males have only one X chromosome onto which the trait is linked. Once that it's affected, then there's no any other chance. Unlike females that can be normal, they can be carriers, and it will take a long time to, for the two X chromosomes to be infected so that they become they suffer from the disease, or rather the trait. A female may have the gene for hemophilia and not show the trait because the normal gene is dominant over the gene for hemophilia. So in that case, we can now see that females have got higher chances of survival than the males. Such females are referred to as carriers, as in this case that we are talking about here. This is a carrier. But phenotypically, this person is normal. But genetically, the person is a carrier. So if the carrier female offspring will be carriers while the other half will be normal when we now cross it. So half the males will be normal and the other will be hemophiliac in that case. Another characteristic that is in human beings is color blindness. So we have what we call red-green color blindness. A red-green color blindness is caused by a recessive gene which is also linked on the X chromosome. It is inherited in the same way as hemophilia. So when we look at males here, in other words, the color blindness is more common in males than females. 1 in every 10,000 of males will suffer color blindness. Less females will suffer, that is 1 in 100 million can be affected. It is the inability, for example, this color blindness is the inability to distinguish between red and green colors. That is in human beings. You may find that somebody is seeing red as green, another one sees green as red. So color blindness is always caused by a recessive gene which is linked on the X chromosome. So if you look at what we are trying to talk about here, then we have seen how these traits appear in these individuals, and I believe now you can be able to relate and understand how they happen. So with that, we come to the end of our lesson of today. Thank you for listening and welcome next time.